Hello and welcome. I'm Gwen Taylor, Senior Developmental Editor and Webinar Program Manager at John Wiley & Sons Publishers, and I'm delighted to introduce today's webinar titled The Complex Pharmacology of T-Cell CARs. This webinar is being co-sponsored by Invitrogen and Current Protocols. Invitrogen flow cytometry is focused on engineering a comprehensive suite of tools for accelerating discovery of new biological insights. From their extensive flow cytometry cell health reagent portfolio and Invitrogen's eBioscience antibody conjugates, to their flow cytometry and instrumentation, they offer an unmatched combination of innovative technologies and premier support for solving complex analytical challenges and for increasing lab productivity for life science researchers. Current Protocols is in its 31st year and provides the highest quality, peer-reviewed, and regularly updated step-by-step research protocols to life scientists worldwide. With 18 titles and over 18,000 protocols containing exquisite procedural detail, extensive troubleshooting tips, and example results, Current Protocols is part of Wiley Publishers. During today's program, we encourage you to submit your questions throughout the event by clicking on the Ask a Question box at the bottom of your screen. Your questions will not be seen by any of the other attendees. The webinar will be recorded and available for viewing in the next few days, and we will send you an email with details on how to access the on-demand webinar. So now it's my pleasure to introduce today's speaker. Charles Prussak holds both PharmD and PhD degrees and has over 25 years' experience working in academia and the biotech industry developing cell-based therapeutics. Dr. Prussak is currently the director of the Cell Therapy Translational Laboratory at the University of California in San Diego, whose purpose is to generate translational cell-based therapeutics that take advantage of breakthroughs discovered in UCSD research laboratories. Dr. Prussak's long history in developing gene therapy products and cell-based immunotherapies spans work in companies including Viagene, Molecular Medicine, and Trajan Therapeutics, several of which he either co-founded or led as CEO. So let's go ahead and get started with a very warm welcome to you, Dr. Prussak.
Gwen, thank you for the introduction. In the next 40 minutes or so, I will be giving an overview on chimeric antigen receptor modified T cells, also known as CAR-Ts. Now, in terms of objectives, upon completion, you should have a greater understanding of CAR-T cells from their generation and how the production process impact their activity and create the complex pharmacology observed for these autologous products. I will review the clinical activities of the two approved CAR-T products, tisagenlecleucel-T, which is also known as Kymriah, and axicabtagene ciloleucel Yescarta, and approval endpoints for these highly active immunotherapies. Along with their profound activities, I will also highlight the unique toxicities that accompany the use of these immunotherapies. Finally, I will go into the future of CAR-T cells to target solid tumor cancers. But perhaps there is no better way to begin this presentation than to recite a 19th century poem from an American poet, Henry Wadsworth Longfellow, entitled, "There Was a Little Girl." There was a little girl who had a little curl right in the middle of her forehead. When she was good, she was very good indeed, but when she was bad, she was horrid. Now, in our enlightened times, there may be major issues in calling a little girl horrid. I'm sure social media would be ablaze, and there would be a great deal of comment in this poem, and the author certainly would be pilloried. So it's not something that would happen nowadays, but Longfellow probably also had issues when, one, he acknowledged this poem was his, and probably more important, he said the poem was about his own daughter. I expect that his wife had something to say about the poem, and of course, his daughter had something to remember her father for the rest of her life. He probably wishes he never had written it. But Longfellow's domestic issues aside, his poem could also have been about CAR-T cells. When they work, they have amazing activity, including the generation of a high rate of in patients with treatment-resistant leukemias and lymphomas who have limited and few therapeutic options, poor medical prognoses, and short life expectancies. And these responses are durable to the point of achieving what we could say are curative responses. And of course, we got to be careful when we're using the word curative in cancer. At the same time, these responses are achieved in the presence of some potentially life-threatening adverse events, which require aggressive medical interventions, many times in intensive care units, and we'll certainly go through that in this presentation. So what is a chimeric antigen receptor modified T cell? Perhaps the best way to define these cells is to visit an immune synapse. This cell will be a cancer cell. Now, here is a simplified immune synapse. And immediately, if you're an immunologist or even had one semester in immunology, you'll immediately notice two things are missing. Cancer side, the blue cell, an MHC molecule is missing, which is usually shown presenting a peptide antigen on its surface. And it's in its place, we have an antigen that the T cell is directly targeting. And on the lymphocyte side, the red cell, we're missing the T cell receptor complex. And it's in place we have a CAR, the chimeric antigen receptor. The chimeric molecule, and we know that because there's 5 different colors here. A chimeric protein is comprised of a single chain variable fragment, also known as the scFv, shown in red on this slide. And this is the business end of the molecule, made up from the light and heavy chains of a monoclonal antibody, a single arm, let's say, of an IgG molecule. It binds the target antigen, and in so doing, sends a signal to a connecting hinge region that also serves to extend the scFv away from the T cell. A transmembrane domain, shown in yellow, which transmits a signal into the interior of the cell, which triggers the co-stimulatory domains and the activating zeta domain in pink and blue. Now, this activating zeta chain, which is usually the signaling moiety on the other end of the T cell receptor, and when we activate these intercellular domains, this results in a highly active cytotoxic lymphocyte that will kill the targeted cell. Now, in creating this Frankenstein monster, in certain ways really a targeted killer, we're creating a lymphocyte that is different from cells employed in the normal adaptive immune process. And maybe at this time, it's important to briefly review how the normal immune system functions. So in the next slide, now normally an immune response is generated in a highly controlled and specific manner. You have the interactions between a wide variety of cells that are part of the innate and adaptive immune system, and then have direct cell-to-cell interactions. And they're also generating soluble ligands that control and modulate the immune activity. Now, imagine if you have a splinter in your finger. Your finger will begin to hurt, more than even getting stabbed with a piece of wood. But you don't really get a systemic response from splinter. When you pull it out, the wound resolves pretty rapidly. The pain goes away. For the most part, you're none the worse for wear. Now, along with this highly controlled immune response, the immune system also has an exquisitely designed and exacting system to design self versus foreign antigens. In utero, and by the time we're born, this system is more or less intact and waiting for additional training. Now, in making a CAR, we are generating immunoreactive cells that work independently of this system. We're bypassing the normal homeostatic control systems. Furthermore, when the T cell CARs finds and binds its targets, we're jamming the system up. This is like playing Beethoven's Fifth Symphony with horns and bass. We could generally get the general gist of what's being played, but it certainly wouldn't be very nice. But this whole process of jamming on the immune system may be needed because the normal immune reactivity against the cancer is broken. The immune system no longer recognizes it. In most cases, the cancer is related to large amounts. In cases of chronic lymphocytic leukemia, you may have kilogram amounts of the tumor. And so we really need to activate and respond against them. And finally, in the case of almost all T-cell CARs, we're targeting self-antigens, which most cancers are generated from self, and this makes them very difficult to treat. This is particularly true when we're talking about CD19. Many ways the CAR products we are generating are robo-killers that will attack any cell that contains the antigen we are targeting. And this is critical because, again, if we're going after self-antigens, we've got to be able to break through this immunoprotective status that our own cells have. Now, in the next slide, we can see the products that have been enrolled in clinical trials. Now, this slide shows a generational evolution of the CAR T constructs over the past 15 years. And you can see the general construct of these molecules have remained pretty much the same over this time period, the only difference being in the number of co-stimulatory domains that have increased from 1 to 2. And in recently looking at clinical trials, I saw some even claim to have a 4, have 3 or more of these co-stimulatory domains. So we can see that in this slide as the light blue areas and the areas below that. And then below that is the CD3 zeta chain. Now, of interest, the 2 CAR products that have recently been approved are second-generation CARs that were described over 20 years ago. One employs a co-stimulatory domain of CD20, CD137, or 41BB. In fact, if we review the history of CARs, we must also review the history of gene therapies, and we can see that in the next slide. Now, on the slide, there's 2 timelines, and we're showing 10-year increments. On the top is the major events from clinical development of gene therapies, and on the bottom with the arrows are the major technical developments that have been required for these CAR therapies. Of course, if we were talking about immunotherapies, we could go all the way back to Coley's antigen use of bacteria to stimulate an immune response against cancers. And he did this work back in the late 19th century. Putting bacteria into patients' cancers, he was actually able to see some of the tumor regress. And certainly this has been the basis for a lot of the ongoing activities for immunotherapies we're currently looking at. However, I'm going to fast forward to more recent times. And gene therapies date back to the 1980s, and the initial clinical trials almost always use extraction of cells, and those began in the late 80s. And as we can see on our time slide, the first gene therapy product was a markericide that Stephen Rosenberg conducted at the NCI. And all these initial gene therapy trials were all using ex vivo products. And that's important because almost immediately after we began using them, I think the people that were funding these efforts started to think about how costly it was going to be to make an ex vivo gene therapy. And I know at my own company, Viogene, we converted from an ex vivo immunotherapy product after treating the first 4 patients to a direct injection. And probably the product didn't work as well, but by direct injection, we were able to make a product that we were able to commercialize in a fairly straightforward way. Now, gene therapies in the 1990s were all driven by hyperbole, and the promises were just tremendous. Monogenetic diseases were going to disappear. Cystic fibrosis and muscular dystrophy weren't going to occur anymore. I think in 1996 there was an Olympics, and a top was using gene therapies to create super athletes. And of course, when these clinical trials started, these great results were not achieved, and in fact, not a lot of activity was really seen. But we did see certain activities, and that was all the way until we got to the late 1990s. And a young man that was 18 years old enrolled into a clinical trial to treat individuals with ornithine transcarbamylase deficiency. Jesse Gelsinger had a mild form of the disease, and he was controlled by diet. And though I believe his disease was progressive, he was going to need a liver transplant at some point. It was be something that was life-threatening at that point. He enrolled in a clinical trial in which he received an adenovirus to treat this disease, and shortly after receiving this product, he died. And this sent shockwaves through the gene therapy community. I received a phone call from a friend of mine working at a gene therapy company a day or so after Jesse died, telling me about this event. to think about using phones to communicate. Certainly that's not the case anymore. But when Jesse Gelsinger died, there was a tremendous upheaval, and I think at that point there was a realization that these gene therapies were not going to be the curative agent. And there was a lot written about the nature of these products, and perhaps they had been advanced too fast out of a research laboratory, and we needed to go back and work on them. And the company I was with at that point, we lost our major pharmaceutical partner about 5 months after Jesse died. And our company lasted a couple of years, and there was just a whole host of other gene therapy companies that disappeared at that time. And our colleagues in academia also had difficult times in getting funding. It was an end of an era. That was the end of the first 10 years and the major hype of gene therapies. Now, after this era, we moved into a relative low-key period, and the gene therapies returned, for the most part, to their roots, which were ex vivo treatments. Don Cohn continued his studies, developed treatment for ADA deficiencies, and a couple of gene therapies were approved for micro-orphan indications. Academic labs at that time went back to developing ex vivo gene therapy treatments, and by the end of the decade, so I guess we could call this the decade of the aughts, a number of academic labs at the NCI, Pennsylvania, Memorial Sloan Kettering, and Fred Hutchinson Cancer Center published promising results with lymphocytes transduced with CAR molecules targeting CD19. And then the next event occurred, which brought gene therapies back to the front again, and these are now called immunotherapies or cell therapies. Nobody wanted to use the word gene therapy, and certainly there were no gene therapies out there. Emily Whitehead was a 6-year-old girl with treatment-resistant precursor ALL. Her amazing story has been the subject of numerous articles, was also a feature of a number of national television shows, including, I believe, 60 Minutes. She received a diagnosis of ALL, acute lymphoblastic leukemia, at age 4, and after her first round of chemotherapy, she achieved a complete remission. The disease was molecularly undetectable. A cure. That's why we have to be cautious about using the cure word with cancers. However, she had a relapse 17 months after her first chemotherapy, and she was treated again. And after reinduction chemotherapy, she had a second remission. But this one was short-lived and only lasted 4 months. And her leukemia at this point became treatment-resistant. She was going to die from her disease. At this point, her parents were frantically searching for a potential treatment. At the same time, Carl June was in a clinical trial at University of Pennsylvania of a CD19 CAR for the treatment of adult CLL, and he achieved and received some really promising results for this treatment. Based on these results, and Emily's parents heard about them, they sought out this treatment, and because of her dire condition, Emily was enrolled in a clinical trial on a compassionate basis with a CD19 CAR. So the CD19 CAR was made for her, and she was treated with this at Children's Hospital Philadelphia. And after receiving the product, really nothing happened after the infusion. There was no toxic effects. But by day 3, she started to develop a low-grade fever that progressed to a high fever by day 4. And on day 5, she was transferred to the pediatric intensive care unit. At this point, this whole process was really starting to get under control. This was followed by rapid progression to respiratory and cardiovascular compromise. She needed to be supported, and so she was put on mechanical ventilation, and her blood pressure needed to be supported. The treating physicians at this point pulled out steroids, which usually stop all immune reactions, but nothing happened. I suspect they also probably tried to use anti-TNF therapy as well, but nothing stopped her reaction. Her temperature hit 105 degrees, and there are pictures that show her on the ventilator, unconscious and swollen almost beyond recognition, surrounded by friends and family who had come really to say goodbye. So, this was the end. They really didn't know what to do. The clinicians, desperate for an answer, analyzed her blood for drivers of her dire condition and came across extremely high levels of interleukin-6. Now, fortuitously, at least for Emily, one of the treating physicians had a family member with rheumatoid arthritis who was being treated with an anti-IL-6 monoclonal antibody, tocilizumab. And with this knowledge, the treating pediatrician gave a dose of tocilizumab to Emily, and her symptoms almost immediately improved. And she was soon taken off a respirator, and her condition soon stabilized. Now, Emily recently celebrated her 12th birthday, still in remission, so that's almost 6 years ago. The result Emily achieved was widely heralded. Gene therapy, which again was now known as immunotherapy or cell therapy, was again on the vanguard. And so a gene therapy was finally going to be approved, and it was this past August. Now, at the same time she had her response, the early clinical successes at the academic centers coalesced into a number of companies who were able to go out and raise hundreds of millions of dollars. One of these companies, Kite Therapeutics, within the last few weeks had their CD19 CAR approved, and I shouldn't say it was Kite that got it approved, because Kite was purchased by Gilead, which purchased Kite for $11.9 billion. And we will review this approval for the Kite product, or I should keep on saying Gilead product, and also for the Novartis product that Emily received later on in this presentation. And I often wonder what the impact would have been if Emily's response was not what it was. In a way, gene therapy now had an inverse bookends with Jesse and also Emily. So, in one case we had an ending and the other a beginning. So if we can go to the next slide, enough of history. Now let's make our own car since we're ready to do that. Now on the slide is a universal process for making an ex vivo gene therapy product. And I retrieved this product from my digital archives, more of my junk pile. It was over 20 years old. The concept is very simple, and this is one that you're going to see over and over again. The process starts with the patient, and again, these are autologous products, and end with the patient. So we go out with the right arm and put it back in the left arm in a way. It takes time to make a full circle, and it's dependent on how much manipulation the autologous cells are required. For CAR T cells, this process takes around 2 to 3 weeks to make the product, and maybe a little bit longer to re-administer the product to the patient. Now, the title of my presentation is The Complex Pharmacology of CAR T cells, and the activity of these agents are driven by how they are made and how these autologous cells are processed, which is outlined in the next slide. Now, let's dive a little deeper into making a CAR T product that we intend to use to treat a patient. Now, if the devil is in the details, I should have put some little flames at the bottom of this slide. I'm going to go give an overview of this process, but almost everything we talk about can be manipulated, and it will change both the quality and the activity of the product that we generate. Well, a good place to start is always at step 1. And to make the CAR T cells, we need to first isolate lymphocytes from the patients that we're going to treat. To do that, we use a process called leukapheresis, which enriches the lymphocytes from other cellular blood components. And these are primarily red blood cells by a process known as elutriation, where cells are collected by their relative density. This is a somewhat invasive process and is conducted in a specialized facility. And we'll hear that in the Gilead product, that there were some patients actually that had this treatment, they did not get their CAR T cells. So once the CAR T cells are collected, they are then transferred to a cell processing unit, step 2. The recently approved CAR T products are sent to central labs, central processing centers where all the products are made. Now, inside these processing centers, the lymphocytes are thoroughly purified. This can be done by employing density gradients like Percol, which further enriches the lymphocytes, or you can completely purify these lymphocytes by employing magnetic beads that use monoclonal antibodies to separate the lymphocytes from other blood constituents. Now, following the isolation of T cells, you're prepared for transfection or transduction, which is step 3. Now, even though transfection using DNA or RNA has been used, we transduce product. So, and I know that there's certain systems out there right now, CRISPR-Cas9, which are people are interested in, but the vast majority of CAR T cells are produced using a replication-deficient retrovirus, and this is either a gamma retrovirus like a pseudotype murine leukemia virus, or a human lentivirus that stably transduce these cells. Products have been approved. Both one uses a gamma retrovirus, the other uses a lentivirus. Now, prior to transduction, the cells need to be activated, and this activation process can take many forms. Perhaps the most used system employs antibodies, and these are monoclonal antibodies that are bound to beads to mimic normal antigen-presenting cells. These antibodies may be directed towards CD3 by itself, or you can include CD28 and CD137 with it. And the lymphocytes are cultured with these beads in a defined media that can contain various amounts of activating lymphokines like IL-2, IL-7, or even IL-15, or combinations of these. At this point, the cells are now activated and can be a retroviral product. Okay, now the transduced cells are incubated with the virus, and we're ready to make our CAR. And these cells can be expanded as necessary using the beads and cytokines that we used before, the cocktail. And using these standard cocktails, these lymphocytes will undergo up to 8 to 10 doublings. And this is step 4. The greater numbers of lymphocytes can be expanded by co-culture with irradiated cells. And large expansive lymphocytes can be attained, and you can expand these cells upwards for weeks and weeks and get a lot of cells. Following transduction expansion, and as needed in step 4, the CAR T cell can now be collected and formulated in a DMSO-containing solution and frozen prior to use. In fact, they can now store liquid nitrogen and can be stored there for as long as necessary before they return to the clinic where the patient is to be treated. And I said as previously, this whole process can take anywhere from 3 weeks up to 2 months. At step 5, the CAR T cells are now ready to be readministered to patients. And now prior to administration, we have one more big step that needs to be done. And to maximize the survival and expansion of these cells before we put them back into the patient needs to have a preconditioning regimen that includes a chemotherapy cocktail that includes the anti-leukemic agents cyclophosphamide and fludarabine. We're actually going to go ahead and kill some of the white blood cells, or not leukemia cells, but actually white blood cells in the patient. This lymphodepletion is thought to be necessary to facilitate T cell expansion and persistence. So really we're giving them a niche for them to get into. And probably more importantly, because homeostasis is going to put a call on to make more white blood cells to fill up this niche, we're probably also providing homeostatic cytokines such as IL-15 to our CAR T product. I can tell you in vitro, if you cold turkey these activated cells away from the activating beads and cytokines, the cells undergo a rapid apoptosis, and really it's a post-activation apoptosis, and the vast majority of the cells die within a short period of time. Now that we know the process to make our own CAR T cells, let's go ahead and make one. So let's look at the next slide. Okay, now for our studies, we start with a leukapheresis product from a donor. We're not dealing with a patient that has leukemia in this case, and these donors are typically giving cells for platelet collections, and platelets are needed a tremendous amount. And the lymphocytes that we have are byproducts of this process. So we get these products from their blood bank, and they're full of a lot of red blood cells that come to us pretty red. And we take this lymphocyte-containing product and further purify it by Phycol gradient. Now, to see what we've actually received from the blood bank, we do flow analysis. And in the left-hand plot, the forward and side scatter dot blot is shown. In this blot, it doesn't really look like we have a purified product. There are a lot of contaminating cells, and these are primarily red blood cells, and also some granulocytes, neutrophils, contaminants. Now, we've drawn a gate around what is classically the area where lymphocytes are found, and these are small 8 to 10 micron cells are not that dense. Above the diagonal are probably monocytes. Now, from this forward and side scatter gate, we can now look at our lymphocytes in a sample by looking for CD3 positive lymphocytes and CD3 negative cells, primarily B lymphocytes or NK cells, and a little bit of monocytes. So if we go ahead and concentrate on the CD3 positive gate, we can now take a look at the CD4 and 8 population. And in this case, we've got a ratio of about 2-to-1 of the CD4s over CD8s. And this is pretty much a standard result we got. So what this means is that we have a lymphocyte population that is normal, and we're ready to activate our lymphocytes prior to actually transducing them. This next slide, we are showing the activation of the lymphocytes that we have. Now, this can be done in a variety of ways. We can use a defined media supplemented with IL-2 that contains CD3 and CD8 activating beads, and this mimics the synthetic antigen-presenting cells that will activate these lymphocytes. They think that now they're actually supposed to be targeting a particular antigen. And when we incubate the lymphocytes in this cocktail for at least 72 hours, we see that a lot of things are going on. So at the top, we can show at day 0 the activation. In this case, we're looking for HLA-DR. And we can see at day 0, there's not much HLA-DR expression. And after the 3 days of activation, you can see that the activation markers are really gone up. And so there's a major shift in HLA-DR expression, but you can use other activation markers like CD25, 69, PD1. And at the same time, you don't even really need to go that far, because by 72 hours, our little 8 to 10 micron cells have now increased to 10 to 13 microns. And you can actually see this by eye under a microscope, and certainly by our cell counter that provides average cell size. So these cells are really activated. They're ready to be now transduced with our viral vectors. So in the next slide, now that we have activated cells, we can successfully transduce them with our retroviral vectors. In this case, we are using a lentivirus that expresses the antigen ROR1. Now, ROR1 is important for fetal development. It guides the development of the fetal nervous system, and we can also see it in vessel formation, skeleton, things like that, where there are fine features with that. Now, during human development by the 2nd trimester, this protein disappears, hopefully forever. And for some reason now, we're starting to detect it in a wide variety of hematological and solid tumor cancers, but not in normal adult tissues. So for some reason, these cancers are using it. As a result, this protein is an ideal target for our CAR T cells. Now, we are using lentivirus, and we use that because these lymphocytes are tremendously very sensitive to lentiviral infection, and we can use very low MOIs. And we're typically using MOIs of 1 and 3, and that's shown in this slide. On the top, again, we have our activated cells, and we have both CD8 and CD4 populations. And we can show at an MOI of 1, after 18 hours, we're starting to see some transduced cells. And by using an MOI of 3, the amount of transduced cells we get are going up and up. And we must be careful at this point that the doses we use, because a greater amount of virus that we use will also increase the potential for a virus to insert into a part of the lymphocyte genome, may activate these cells to the point they replicate out of control. Now, transduction-based leukemias have not been detected in any of the CAR T trials to date. And again, these are using gamma retroviruses on lentiviral vectors. So this is good news, but this is something that you don't really want to push. So now we have a nice expression of Aurora CAR on the T cells. Let's go test it. Now, when we began these studies, we wanted to measure the activity of our CARs by using the chromium release assay using chromium-51 that we employed 10 years ago. Unfortunately, we don't have a radiation safety license anymore, and perhaps more importantly, no one in our building had a gamma counter. So that really stopped us from doing that, and that forced us to go to a second choice of assay. And we defaulted to flow assays. We thought that would be a good way to go. And initially, we thought about using the usual cell viability assays like Annexin-5, DIOC6, PI, those things. Well, we soon realized that we're not dealing not only with apoptosis, but also necrosis. And with our robo-killers, it was important that we were able to account for all the cells that we were targeting. Therefore, to simplify things, we used cell labeling dyes like CSFE and a variety of cell traces. And so on this slide, we've taken a ROR1 positive JACO cell, which is a leukemic isolated from a patient with a transformed mantle cell lymphoma. Again, this is a B lymphocyte. And we've incubated them with our ROR1 CAR T cells after labeling them with a yellow cell trace. And in the left forward and side scatter plot on the top, you can see two populations of cells. And we have to open our gates to analyze them. Even our little lymphocytes now are running bigger, but it's quite obvious that we have two cell types. And if you look at the bottom part, you see the labeled cells, which is our targeted JACO cells, are been labeled, and 50%, 42.5% of the cells are labeled with our yellow cell trace. Now we incubate them with our CAR T product, our ROR1 CAR T product, and after 43 hours, we initially show, again with side scatter and forward scatter plots, that these cells are disappeared, and we only see one population of cells. And as we can see below, now our JACO cells have disappeared. They've been killed. Now, as a control, we have to run a second culture with only activated cells, and this is an optimum because the two cultures are really not equivalent. They are separate, but not equivalent. But since we're using cell dyes, it's pretty easy to go ahead and use multiple cell traces to co-incubate cells and see what we're doing with a CAR T, and that's what we see in this next slide. So now we're using the same assay using two separate dyes. In this case, we're using a cell line called MEC. One of the MEC cells we have transduced with a ROR1, so this is ROR1 positive MEC1 cells, and then we have the ROR1 negative, the normal MEC1 cells. And so again, we can see in this plot, now we have an E to T ratio where we have 3 effectors to one of the ROR1 positive cells, shown in red, and one of the ROR1 negative cells, shown in the yellow. And again, if we look at after 20 hours, and we again look at it, now by co-incubating these cells, we are now getting specific killing of the ROR1 population, shown on the right, while our ROR1 negative cells, for the most part, are being left alone, and they're now expanding out. So this was pretty nice. We can now differentially kill a target cell in a population of other cells. And as we show on the next slide, this process is both dose and time dependent. So now we've gone back to look at both our MEC1 negative cell lines and our JACO cells and our MEC1 ROR1 positive cells, and looking at a variety of effector to target ratios and looking at a time-dependent method, we can see that these cells do work in a time and dose-dependent manner. Now, if I took this ratio up to 10 to 1, now if we increase them up higher, the target cells will get killed really a lot faster, but we'll also begin to see a major killing of the ROR1 negative cells. So we're really showing you the fillet of results here. And we are generally dealing with an allogeneic response. And depending on the donor product, many times we'll get substantial killing with our non-transduced and therefore our non-CAR expressing cells. And so these activated lymphocytes are very much like lymphokine activated killers have been described before. So fortunately, we have access to patient-derived xenografts and mouse tumor models, which we have also access to autologous lymphocytes, and we're currently testing these systems with our ROR1 CAR T cells, and this should give us a cleaner picture, specific killing. Now, the result from this slide is from leukemia cells. We also see the same results with solid tumor cancers, and just briefly, I'm just going to show you this. So on this slide, we have 2 head and neck cancers that are being co-incubated. One of them is a ROR1 positive, and this is stained green. And a second cancer is ROR1 negative, it's stained red. And the panel A, we see the co-incubated of these 2 cell types. So you have both green and red cells. And this is at time 0. And in B, this is the same culture at 44 hours, and we can see these cells are really packed in at that point in time. Panel C is where we've added now our ROR1 CAR, and at a 3-to-1 E to T ratio. And you can see the green cells are starting to disappear. And by 44 hours, it's very difficult to find any green cells, and the red cells are packing together. You also show clear areas, though, and because we're also getting non-specific killing. So, again, this is a particular problem with these in vitro assays. And, if we're using autologous products, we'd expect them to be a lot cleaner. And we're now also testing our PDX models where we're using autologous products, and hopefully that'll give us cleaner products. But all we can say is that we're getting a certain percentage of ROR1 specific killing, and this varies between the donor products employed, okay? So from that point, and we've gone ahead and tested this in vitro, let's go advance now to clinical studies. Now, in the time remaining, I want to review the clinical studies that led to the approval of the 2 CAR T products in the last few months, tisagenlecleucel, which is also known as Kymriah, for pediatric precursor ALL, and axial-cartagene sialylysyl, which is known as just CARTA, for adult patients with diffuse large B-cell lymphomas. Of course, the clinical sites of these programs have stimulated the field. Now there are hundreds of clinical trials being conducted and investigating these activities throughout the world. Now, where do you think all these clinical trials are occurring? Now, according to clinicaltrial.gov, surprisingly, only in 2 countries, China with 108 clinical trials and the U.S. with 95. Great Britain is next with 9, and the rest is spread over the world, primarily as sites for large Kite, Gilead, and Novartis pivotal trials. So now these clinical trials, the vast majority of them now are investing CAR T cells for the treatment of leukemias and lymphomas. I guess success breeds imitation. And, we're also using new approaches to target the CD19 and the other usual hematological cancer targets, including CD30, 33, and 38. And probably greater than 80% of these trials are targeting liquid tumors. Well, the driver of all these clinical trials are the approved products. And the first approval was the Novartis CAR T that targets pediatric ALL, the disease that Emily Whitehead had. And tisagenlecleucel is known by the trade name Kymriah. Kymriah is a CD19 CAR indicated for the treatment of patients up to 25 years of age with B-cell precursor acute lymphoblastic leukemia that is refractory or in second or later relapse. This is the disease that Emily Whitehead had. There are about 35 to 4,500 cases of pediatric ALL in the U.S. per year, and 80% of the children will respond to frontline chemotherapy. And those that have a relapse, 10% or so will be treated with Kymriah. I believe that Novartis said they expect to treat up to 600 children with this disease per year. And I don't know if this is only in the U.S., but also includes Europe. For this pivotal trial, a total of 107 patients were screened, and eventually 63 were evaluable for efficacy. Now, a major failure that the patients that were enrolled that did not receive the product were due to manufacturing failure. Now, Novartis says that they have solved this problem in the last 60 or so products. I believe they've been successful. So if we look at this pivotal trial and we look at the overall response rate within 3 months, and we look at the complete response rate or the complete response rate with incomplete reconstitution of the marrow, you have 82.5% of the children responding, which is just amazing. More importantly, if we look at the response with MRD negative bone marrow, means they can't detect the disease, that's also 82.5%, and the duration of remission is not been determined yet because it hasn't been released. And the overall survival, again by Kaplan-Meier estimate, is at month 6 at 88.6%. So these children are living a long time with this, and of course Emily Whitehead is 6 years alone, and we can really now start thinking about cures. So you've gone from a situation where you have an incurable leukemia, which is probably going to very much shorten your life or die from it, and now we're getting to a point where treatment now is curing these children. But this, along with this impressive activity, there's also serious adverse events, which are seen in the next slide. Now, for patients that received Kymriah, 11 of them have died, and 2 of these deaths occurred within 30 days of infusion. 7 of these were disease-related and were attributable to infection. And of the 2 deaths before day 30, 1 patient died with, again, cytokine release syndrome and progressive leukemia. The second patient had resolving cytokine release syndrome, but they had multiple organ failure, and they also had an intracranial hemorrhage and died from that. Now, the impact on the immune system is tremendously severe, and we see decreases in white blood cells, neutrophils, and lymphocytes, and this led to serious infections. And so grade 3 and 4 bacterial, viral, and fungal infections occurred in 24% of the treated patients, and 3 patients died from this. And our good friend cytokine release system occurred in a high number of these patients, and this is even with tocilizumab treatment. And so this is a very serious SAE, and we'll see later that patients with this, a large number of them died. And I also didn't put on the AE table was the incidence of neurological toxicities, which is another hallmark of CAR-T therapy, with grade 3 and 4 toxicities seen in 18% of the treated children, including encephalopathy, delirium, disorientation, confusion, and aphasia. And we'll also see that this is an extreme toxicity associated with CAR-T products. So in the case of Kymriah, we had tremendous curative response. It was also had attendant extreme toxicity. And this is why these products are given in specialized and approved centers. So let's go to the next product, which is the most recent approved product, axicabtagene ciloleucel, and this is also known as Yescarta. And this product was approved on October 18th. And Yescarta is a CD19 CAR indicated for the treatment of adult patients with relapsed or refractory large B-cell lymphoma after 2 or more lines of synthetic therapy, including diffuse large B-cell lymphoma, primary mediastinal B-cell lymphoma, and transformed follicular lymphoma. Now, 80,000 patients per year are diagnosed with non-Hodgkin's lymphoma, and 20,000 will die. Now, in this trial, patients that have failed 2 or more synthetic therapies, systemic therapies, and that's what we have patients who are treating in this trial, have a median life expectancy of less than 6 months. And around 20% of these patients will live 1 year and 10% for 2 years. So it's a very bad disease. These patients have a very short life expectancy. Now, Kite and Gilead expect that 7,500 patients will be treated a year in the U.S. for this. I've heard numbers as high as 35,000 patients that would be eligible for this treatment between the U.S. and Europe. So there's a lot of patients that will benefit from this therapy. So in this pivotal clinical trial, we're also called ZUMA-1, 111 patients were enrolled in the clinical trial, and only 1 did not receive the product due to manufacturing failure. 9 other patients were not treated primarily due to progressive disease or serious adverse events following our friend leukapheresis. Now, multiple patients died in this clinical trial. I don't know how many. At least 3 that we know of, at least have been reported. But the major adverse events seen were, again, lymphopenias and leukopenias, things that impacted the hematological system. And as a result of this, again, these patients become very susceptible to infectious disease. 16% of the patients had grade 3 and 4 infectious diseases, which could be bacterial, viral, or fungal in nature. We also saw a good number of patients with encephalopathy, a little bit more so than we saw with pediatric treatment, and cytokine release syndrome, which may be a little bit less. But again, these are very serious adverse events. And so again, we have a very high complete response rate of 52%. And this is a durable response because by month 6, 36% of those patients were still in a complete response. And as I told you to start, 50% of the patients will probably be dead with this disease within 6 months. So this was really the basis for approval in October 18th, this product was approved. So let's go through just really quickly. I can just also show you that tisagenlecleucel-T, the product that we find in Kymriah, is also being used for adult patients, and they're getting similar type responses that, again, Kite and Gilead are getting with their product, and this will probably be the next one that gets approved. And so just to hurry up, because I've been told everybody I'll be done in 40 minutes or so, we can go ahead and see the death-following T-cell CAR therapies. And these are widely heralded when these occur, but you must realize now that hundreds and hundreds of patients have been treated with these diseases. And, you know, it's something that's probably not that unexpected. And, of course, leading this parade again is cytokine release syndrome. And this is most commonly observed toxicity, and it happens probably in just about all these patients that are treated, but it can progress to a life-threatening situation, even again having access to tocilizumab. We also get a lot of encephalopathy syndromes, and patients have died from these neurotoxicities. And the CAR T-cell related encephalopathy syndrome is known as CREST. And this is the second most common adverse event. And then this can happen concurrently with the cytokine release syndrome or independently by itself. And so this particular graph was generated by a CAR T-cell therapy working group, so-called CAR-TOX group. And this is a number of investigators from multiple institutions and medical disciplines who have experience in treating these patients, and they tried to now put together their combined experience to help people in treating these patients, telling you what to expect, how to treat it, and these type of things going forward. So with that, again, the reference for this CAR-TOX working group is given below. It actually has some good information on how these patients are, what to expect from these patients, and how to treat these serious adverse events. So let's very quickly then progress to solid tumors. You know, after leukemias, we're now faced with a much tougher test, and it's always been the case. New therapeutic agents are always first tried and found to be successful in these liquid tumors, and at some time later, they're advanced to treating solid tumors. Now, these solid tumors are really highly engineered cells created to avoid the human immune system. You're also not concerned with normal homeostatic processes. They can pretty much do what they want. And, you know, they take advantage of that because generally they're not all that genetically stable. They can just about choose any genetic element they want. And if they don't work, they can generate new forms of these proteins and genes and use them to any way that they want to. In many ways, they're vehicles to allow DNA to replicate. And, of course, DNA loves to replicate and make new forms, and that's the basis for all life forms. So with that, this is what we've got to start going. Now, these are particularly tough things to go after because usually, again, they're taking self-antigens and they're using them to their own advantage. So we'll know very much about, there's a great deal of activity going on right now to look at the genetic elements that make up these solid tumors, the genome, the transcriptome. We'll know what's going on with them. But again, making treatments is going to be a long-term process. And pretty much we've got to go back to using the tried and true targets that we've had in the past. And if we go to the next slide, we can see that there's many fewer clinical trials being attempted with the solid tumors. Again, a majority of them are occurring in China. And of interest is, and I think probably the real next targets will be those that are centrally located. For instance, City of Hope has a very promising trial with IL-13 receptor alpha-2 going on, and you can see a lot of other people are looking at gliomas. But people are also looking at other things like HER2, and HER2 is found in a lot of cells. And on one of the early clinical trials, a patient died from receiving a HER2 CAR T because from pulmonary arrest. So we very much have to be very careful when we're using these agents, okay? So very quickly then to wrap things up for solid tumors, there's a number of points that we need to consider when we're making these agents. And first thing we must consider is the target antigen We'd certainly like to have that target antigen enriched on the tumor that we're attempting to treat. We don't want to be going at too much after self-antigens because we're going to get some very dire results. So when we go ahead and choose our target, the next big issue we're going to be facing is delivery of this product to the tumor. And these tumors many times have very tortuous vasculature to them. They're not really concerned with having a really good bloodstream. And many times that works to their advantage because it makes it difficult for the immune system to get them. And we're going to have the same issue of getting our product back into the tumor. And even if we're fortunate to get it back into that tumor bed, tumor microenvironment in many cases is very toxic. So it could be very anoxic. It could have very low oxygen tension. It could be very acidic. And our nice CAR T product that was made in a nice laboratory dish is now got to be able to go ahead and work in this environment. And even if we can do that, then we have the next big issue is the tumor immunosuppressive repertoire, the checkpoints. And this is the primary method by which these tumors are probably evading the immune system. And when our CAR T product starts to get into this tumor bed, it'll probably pass cells that are part of the adaptive immune system. They're there in suspended animation, being stopped from doing their activities by these tumors. So these are a lot of different things that we're going to have to address. And there's a tremendous amount of research that is ongoing right now in this field. I just looked at PubMed.gov and put the search terms chimeric antigen receptor in 2017, and there was over 470 articles that have been written to date. And 2017 is certainly not over yet, and I suspect there'll be over 500 papers. And people are trying all different types of ways to improve the activity of these compounds. So, in conclusion, CAR T gene therapies are now accepted pharmaceutical agents. So after 30 years, finally a gene therapy agent has been approved. The generation of these CAR T products influence the activity of these products, as we've shown. So how you make them will also determine the activity they have and how long these products will remain in the treated patient. And also curative responses with the CD19 CAR cells have been achieved. And, you know, this is going to be expanded, and hopefully at some point we can say the same thing about solid tumors. But these solid tumors represent a major challenge, and we're probably going to have to be developing next-generation CAR T products to get activity in these dire cancers. So at this point, I'd like to thank everybody for listening. Thank you.
All right. Well, thank you so much, Dr. Prussak, for that fascinating presentation. So let's go ahead to the question and answer segment now. So if those of you viewing haven't yet submitted a question for our speaker, now is the time to do so by clicking on the Ask a Question box at the bottom of your screen. So let's go ahead and see what questions have come in so far. First one we have is, how do you prevent lymphocytes from undergoing apoptosis after activation in vitro?
That's an excellent question. In fact, once you do activate these cells, they have one way to go, and that's either to go forward and start to divide and replicate, or once you do take away the stimulation, they do die. So to keep them going, you need to keep them stimulated. That means that you need to continue to provide the activating beads and cytokines, and they will continue to go at least for a few doublings, and then you need to do co-cultivate them with irradiated cells or something like that, and they will progress and replicate for many, many doublings after that.
All right, next question is, you showed the transduction of the CD4 and 8+ cells with the lentivirus. Do you know what subset of cells you are transducing as well?
Yes, we've looked at them, and so we've looked at all the stem cell different markers and things like that, and we have tried to steer the cells, let's say, into memory stem cells, or at least into memory populations. And to this point, we haven't been successful, and we pretty much are transducing the cells right across the board, from everywhere from effector cells through the memory population. So. But we're working on that, and we're trying to steer them. There are some papers suggesting that you can do that, but we haven't had success so far.
Okay, we're actually at an hour right now, but we have permission to go a few minutes over, so we can definitely take a few more questions. So the next one is, prior to administration of the CAR T product, you mentioned that patients have to be treated with a chemotherapy. Could these patients be given cytokines directly to aid in the survival and expansion of the T cells?
Yes, that's another good point. Of course, the chemotherapy is very important for these cells to survive, post-activation. And when you're going into the patient, of course, you're dealing with homeostasis. And when you're killing off certain populations of lymphocytes, you then have a call at that point to reproduce those cells. So where we would use, say, in vitro, certain cytokines, IL-2, IL-7, 15, 21, those type of things. In vivo, those cytokines are used, and probably others along with them, at physiological concentrations. It's something that would be very difficult to reproduce if we were trying to do that artificially.
All right. Next question asks, how long do the CAR T cells stay in the patient?
Well, potentially, and we've seen these in these clinical trials right now, particularly those with the Novartis product. These patients may have these CAR T cells circulating with them for the rest of their lives. And so they can be very long-acting. So this is one aspect, and this is also associated with greater activity, this long-term survival of these cells.
Okay, and perhaps related, the next question is, how long will the patients that receive the T-cell CARs be immunosuppressed?
Well, again, if we're targeting CD19 and we're now at this point, we're going to be taking out normal B cells along with that, patients, again, could be immunosuppressed for the rest of their lives. And so patients receiving the CD19 CARs need to, for instance, get IVIG, so they need to have immunoglobulins. And again, they'll have to be watch very closely for any evidence of infectious diseases.
Okay, the next question is, do you isolate untouched T cells, or is positive selection okay?
Oh, you can. There are ways, and this is, I believe, a technology that came out of the City of Hope. They're using the HER1, EGF receptor product with their lentivirus, and it allows them to go ahead and select transduce cells. We, at this point, are just taking bulk cells. So we're taking everything that goes in, and we're not selecting out our cell populations, either for transduce cells or populations of 4 or 8 T cells.
All right. Next question says, you use a lentivirus to transduce the T cells, but what about gamma retroviruses?
Well, those have also been used, and that goes back to what we were doing, almost 30 years ago. The gamma retroviruses do transduce these cells, probably not as effectively as a lentivirus. This is all the technology that Kite is actually using to make their product right now. So they're using gamma retrovirus. So you can use those as well. The tropism is probably not as great as it is with the lentivirus, but you can use the gamma retrovirus as well to transduce T cells.
Okay, next question says, thank you for your excellent presentation. That's not a question, that's just a compliment. But the actual question is, what are the long-term side effects or adverse events of the CAR T cells?
Well, the long-term effects is that when they go in, they could potentially be there for the life of the patient. And probably, again, the one that we see most often right now is the immunosuppression, because of the loss of your B cell populations, your CD19 populations. And so that is something, if you have different targets, then you can potentially have other issues. Again, these cells can go in and have these activities against targets. And if you're asking about long-term toxicities, we have to think about when we're targeting something that this is a potential with these therapies.
All right, we have time for a couple more questions. Next one is, what do you think of studying patients' T cells at a single cell level to better understand the T cell responses to these therapies?
You can, and realize that we're starting out with whole populations of cells, and so we're really dealing with Poisson-type distributions, and not knowing really what the true effector cells are, and what we're trying to do is hope that we hit something. Again, this technology is very early on in terms of development, and if we look at individual cells, we maybe have a better idea of what the effector cells are. That may be a bit of a difficulty at this point, but I'm sure as time goes through that we'll have a greater understanding about what the particular cells that we need to actually transduce and what properties they need to have.
Okay, next question asks, do you know of any clinical trials or applications that are underway for using CAR-T to treat autoimmune diseases?
That's, that's something right now, and I think that I've run across those. Right off the top of my head, I can't, but I mean, it'd be very easy to do is to go into clinicaltrials.gov and look at the use of CAR-Ts for the treatment of them. If there is something potentially against lupus or those type of diseases, there may be something, again, we may be dealing with B cells that may have some impact on that disease. So I can't recall anything, but it'd be certainly something it'd be fairly easy to find.
Okay, next question is, are these T-cell processing and production steps written up anywhere?
Actually, that's a good question. If you look at it, and this is a little bit of the how these products are put together is a bit of an art and something that's not typically shared. I believe the City of Hope has written up a paper that describes their process that's pretty comprehensive, and it's probably a good place to look at. I don't know what the citation of that is, but I think if you look at CAR-T production and City of Hope, you may find their particular paper.
All right, we're really over time now, so I'd like to wrap up the question and answer segment and let you know that today's webinar has been recorded and it will be available for viewing in the next few days. We will send you an email with details on how to access the recorded webinar on demand. So on behalf of today's speaker and our sponsor, Invitrogen, we sincerely appreciate your attending today's webinar and look forward to your attendance at future events from Wiley Publishers.
Thank you, Gwen.